So let's go. So hello, it's again Thursday, and we have the worldwide seminars for the neuroscience of addiction, the global ones. And today it's the pleasure that I'm here with Marina Picciotto and Cristina Miliano. And without any further ado, Cristina is going to introduce our speaker. She'll have 45 minutes, then we have questions. And don't be shy, ask your questions. Cristina, please.
Yes. Dr. Picciotto received a bachelor degree in biological science from Stanford University, California. She then earned a PhD in molecular neurobiology at the Rockefeller University in New York City, where she worked in the laboratory of Paul Greengard. She then completed a postdoctoral fellowship with Jean-Pierre Changé at the Institut Pasteur in Paris, and she finally joined the Yale faculty in 1995. Dr. Picciotto is currently editor-in-chief of the Journal of Neuroscience. She served many committees, such as the Scientific Council of the NIDA and the Society for Research on Nicotine and Tobacco. During her career, she published almost 240 publications with over 20,000 citations, and she was awarded with multiple awards for her findings on nicotine addiction and the role of acetylcholine receptors in brain function and development. She does represent a role model for all women in STEM, so we're very excited to have her here with us today. And without further ado, I'll give the word to Dr. Picciotto.
Thank you so much, Cristina. That was lovely. So, welcome, everyone. Thank you for joining us today across various time zones. And I'm going to talk today about recent work from the laboratory around acetylcholine dynamics, and particularly in the basolateral amygdala during a reward learning task. So we know that one of the things that drives ongoing use of addictive substances is the fact that all of the drugs that we know that are abused by humans can enhance cue-reward associations. And I'll give you an example from nicotine and nicotine addiction that might illustrate this point very clearly. So smokers who smoke tobacco specifically to obtain nicotine from tobacco smoke start out by smoking a cigarette, and perhaps there's some euphoric action. When I was a teenager, tried it, and in fact, most of the thing that I perceived was negative. It was the coughing and the autonomic dysfunction, which may be why I didn't progress to being a daily smoker. But those who persist through the initial aversive aspects begin to associate the aspects of smoking that allow you to obtain nicotine with the internal states that drive ongoing smoking. So for example, a smoker might see a pack, and before they actually have a conscious thought, that cue, the pack that was previously neutral, now causes a motor pattern, including reaching for the cigarette, lighting it, smoking it, that may in fact be purely unconscious. You begin the process by observing the cue, and then the pattern, the motor pattern of obtaining the drug, in fact, becomes extremely highly likely to occur. Now, this also occurs with flavors and with aspects of e-cigarettes that are extremely attractive to initiation of use of e-cigarettes, for example, by adolescents and youth. I think cotton candy is not likely to be a flavor that is attractive to dependent smokers when they're thinking about using e-cigarettes to quit. Instead, it's a really attractive way to get new users to take the product. So this mildly reinforcing flavor, which is cotton candy, is now becomes associated with the delivery of nicotine through e-cigarettes. And again, now the perception of that sweet taste or that scent of cotton candy may in fact initiate craving and initiate motor action in order to get the nicotine from an e-cigarette. So that's the kind of framework where we began to think about this work. So we know that the basolateral amygdala, from work from many laboratories, and I'm showing you one example from a review in Current Opinion in Neurobiology from Grundemann and Luthi, is responsive to both aversive and rewarding stimuli. So we know that the neurons fire in an unconditioned way to aversive and rewarding stimuli. But there's also a tremendous amount of work on the plasticity in the amygdala to link those unconditioned, aversive, and appetitive responses to previously neutral cues. And I'm showing you here an example from fear conditioning, where initially there are a number of cells in the basolateral amygdala that are responsive to a conditioned stimulus. And then after linking the presentation of a neutral cue to an aversive stimulus, a foot shock, now you can actually potentiate the activity of more of the subsets of neurons in the amygdala, particularly basolateral amygdala, to the conditioned stimulus. So there's underlying basal firing. There's also plasticity in the structure that makes it a very attractive structure to look at for assigning valence and learning about the valence of previously neutral cues. And this is also true in terms of rewarding cues. And this is work from Zhang and Li, where they looked at the ability of a liquid reward in animals that had been liquid deprived to be linked to an auditory cue. And what they saw was that initially there were a proportion of cells that were linked to the neutral cue, and that increased over the course of learning. So there's clearly good evidence in the literature for both learning about aversive and appetitive aspects of cues. So another thing that you need to know about the basolateral amygdala is that it has an extremely potent acetylcholine projection that is reflected here in this slice through the mouse brain, that anyone who has done any work in rodents where they've used Paxinos and Watson Atlas may recognize a picture of this kind. And what you can see is that this counterstain that allows the researcher who's trying to section brains on the microtome to determine where they are in the brain is counterstained with acetylcholinesterase. So a marker of the primary degradative enzyme of acetylcholine. So it is clear that the amygdala, and particularly the basolateral amygdala, is a source of extremely high expression of this degradative enzyme for acetylcholinesterase. Now that may be because it has a very strong projection of acetylcholine from the basal forebrain complex. It may be that the levels of the degradative enzyme are high in order to keep acetylcholine levels low. And so the first thing I'm going to show you is some evidence that in fact acetylcholine signaling in this brain area is essential for setting the overall tone, the overall basal firing of the basolateral amygdala. And so I'll start with that historical work. So before I get into this, I need to give you one more piece of information, which is that acetylcholine signals through two broad families of receptors. So the nicotinic acetylcholine receptors, which are the primary targets in the brain for smoke to nicotine, vape to nicotine. Nicotine binds at the exact same site as acetylcholine, activates, opens these receptors, and then desensitizes them as well. So you have a wave of activation and then desensitization or longer term inhibition of their firing. And then there are muscarinic receptors, which are G protein coupled receptors, and they can either be coupled to excitatory G proteins or inhibitory G proteins. So in fact, they could be on terminals of the inhibitory muscarinic receptors could be on acetylcholine terminals, decreasing tone of acetylcholine, or they could be on cell bodies, increasing firing. And indeed, this panoply of receptors for acetylcholine make it very difficult to predict what will happen when you release acetylcholine into a structure. And overall, what will happen is that there will be an integration of excitatory and inhibitory signals on multiple compartments of neurons in the structure. And here in the BLA, there's good evidence for nicotinic receptors on terminals, muscarinic receptors on terminals of many different subtypes of neurons, also on the cell bodies of principal neurons and all of the major GABAergic inhibitory neuronal subtypes. And overall, this leads to the execution of a neuromodulatory function for acetylcholine, so that overall what happens is that changing the firing of an entire group of neurons of many different subtypes results in a change in state in the structure that is a hallmark of neuromodulatory neurotransmitters. And I don't have time to talk more about neuromodulation here, but I think that the underlying role of acetylcholine is as a neuromodulator to be released in response to salient stimuli, whether they are rewarding or aversive, and then to shift the state of multiple different brain structures into a different program. So resulting in coordination of output of a number of structures that can execute behavioral functions. So here's a piece of data suggesting that acetylcholine signaling in the BLA in particular is extremely important for the overall firing rate of the neurons in that structure. So here we did a CFOS study, and this was experiments done by Jan Mineur, who is a longtime collaborator in the lab. And what he did was to expose animals to a brief stress, and then to section throughout the brain. And what you can see here is the basolateral amygdala, the hypothalamus, nucleus accumbens, et cetera, and to block nicotinic receptors using two different pharmacological methods, one that completely blocks signaling, so a pore blocker, a channel blocker, mecamylamine, and one that is a competitive low efficacy partial agonist. And those of you who are pharmacologists know what this means. For those of you who are not, what this is a drug that interferes with ongoing acetylcholine signaling. So it will actually tweak that receptor. It will open it a little bit, but not as much as acetylcholine. So what happens is that if you have ongoing acetylcholine signaling in this structure, interfering with its signaling with this partial agonist will knock it down. So if you had an agonist that was having an ongoing effect in a structure, blocking its activity with this low efficacy partial agonist would decrease its efficacy. And that's what he saw particularly in the basolateral amygdala. So you can see that the full antagonist of nicotinic receptors, and this is across all classes of nicotinic receptors, decreased CFOS expression pretty much everywhere that Jan looked. But particularly in the basolateral amygdala, he saw a very large decrease in activity of the structure as reflected by CFOS. And it was also exactly the same if he interfered with ongoing acetylcholine signaling by giving this partial agonist. So this suggests that indeed the tone of the BLA is particularly sensitive to nicotinic stimulation, and the structure is getting a broad activation as a result of acetylcholine inputs. He also confirmed that this was occurring directly through acetylcholine signaling in the BLA. All of the pharmacological experiments were done with peripheral administration, but locally in the BLA, he could use a molecular genetic method, and that is to deliver small hairpin RNAs that specifically targeted different subtypes of the nicotinic receptor family. And those small hairpin RNAs found their cognate RNAs. After a few weeks, we saw a decrease in the RNAs encoding these nicotinic subunits, and then a decrease in their activity. And what he saw is that chronically, he saw the same thing, that if he knocked down the primary heteromeric, the primary high affinity subtype of the nicotinic receptors, he saw a significant decrease in CFOS that was due to acetylcholine receptors in the BLA itself. And this occurred not just acutely, but across many weeks of survival. And the same was true to some extent, although less so, with another subtype of the nicotinic receptor family, suggesting it was a fairly broad effect across nicotinic receptor subtypes. So first conclusion is that acetylcholine does act through nicotinic receptors to contribute to the baseline excitability of the basolateral amygdala. It's important to have this acetylcholine input. So then the next question is, when does acetylcholine actually get released in the structure? And here the question is around cue reward learning. Can we find a change, or can we actually identify release of acetylcholine during the various phases of a cue reward learning task? And this is work largely done by Rick Krause in the lab when he was a graduate student. He's now defended his PhD successfully, even during shutdown, and is now a postdoc in the lab. And so let me actually back up a little bit, and that there was a very nice experiment, a set of experiments done by members of the Lorna Roll and David Talmage's lab, where they showed that acetylcholine, and particularly nicotinic acetylcholine receptors, were important for a different kind of emotional cue emotion learning, and that is in fear conditioning. And what they showed using a combination of optical stimulation, optical inhibition, and blockade of particular subtypes of the nicotinic receptor was that if they increased acetylcholine signaling during the pairing of a neutral cue and a foot shock, they could delay or decrease extinction of that fear learning. Of course, under normal conditions, animals learn to associate a cue with foot shock and then over time will extinguish that learning. And if they inhibited the acetylcholine input to the BLA, then they could impair acquisition of that cue outcome learning. And of course, then extinction was more rapid. So overall, this suggested that there was a role for acetylcholine, particularly working through the alpha-7 subtype of the nicotinic acetylcholine receptor to enhance plasticity in the structure and to enable the cue, in this case, cue foot shock learning to occur. So the first question that Rick asked was, now we know that tonic ACH in the BLA is important for baseline activity of the structure. We know that acetylcholine contributes to plasticity underlying fear conditioning. What about now cue reward learning? Is phasic BLA acetylcholine a more general learning signal that might be able to contribute to cue reward learning? So what Rick did was to set up a task where we could get operant responding for a reward. And in this case, the cue was a tone, and that when that tone was played, then their active nose poke was available. If the animal poked its nose, then ensure, which I have to say in full disclosure, I have tried and is completely vile, but mice love it. It's a sweet, highly caloric drink. When they poke their nose during the time that the cue is being played, the Ensure becomes available. They can come across, drink the Ensure, and get a reward. Okay, so there are multiple phases of this task. There's a 30-second variable interval between trials, so the animals can't just learn the timing. They have to pay attention over time to the cue. But in different phases of the task, they acquire different aspects of this training. So first, they're food restricted so that they're very motivated to work for the reward. They're handled so that they're very calm. They're familiarized with the apparatus so that they know that nose pokes are there. They're not freaked out by the novelty. And then we have a phase that's called pre-training. And in the pre-training phase, that cue is presented. They only get the reward if they nose poke during the cue. However, there's no punishment. So they can nose poke as many times as they want. And as long as they have been nose poking when the cue is available, they'll go across and get the reward. Then Rick changed the contingency. So during training, now if they nose poke during the time that the cue is playing, yes, they get the reward. But if they nose poke when there is no cue, now they get a timeout. And so they're not able to, there's change in the lights, there's clearly a difference in contingency, and now the reward will not be available when they nose poke. So during that training, they actually have to relearn to withhold their nose poking in order to appropriately respond to the cue, and they definitely have to use the cue. And then there's extinction learning where everything is the same, but no reward is presented. So this is the same task I will be talking about for the entire rest of the talk. And now I want to introduce a wonderful tool that has revolutionized the work in our lab. It is one of those tools that is ideal for a neuroscientist because we were waiting for it to happen. For many, many years, we have been using pharmacology and molecular biology to manipulate the cholinergic system. We've tried many different ways to measure acetylcholine during the behaviors that we're interested in. We've done microdialysis. We've made our attempt at amperometry. But the problem with many of the other methods is either that they are very slow, so microdialysis is on the minute time scale, or that they are difficult to get to work. For example, the amperometry that we were not able to get to work, although there's beautiful work from other laboratories, including the Parikh and Sartor groups. But this new method has been really transformative. And so Yulong Li and his colleagues have developed a series of fluorescent neurotransmitter sensors. And in particular, we've been using an acetylcholine sensor that is based on one of the families of the acetylcholine receptor families, so the muscarinic family. And in this particular construct, the G protein coupled receptor is intact. All of the transmembrane domains are the same. The acetylcholine binding domain is available, but the internal signaling portion of this receptor has been replaced with a permutated GFP. So now when acetylcholine binds to this G protein coupled receptor and changes its conformation, it alters the structure of GFP. We get release of photons. And if we put a fiber over the brain area in which this sensor is expressed, we can measure the fluorescence from this construct over, as far as we can tell, the lifetime of an animal. So this is packaged into an adeno-associated virus. It is infused into the brain area of interest, in this case, the BLA. We put a fiber over the area where we want to measure the ACH levels, and then we can perform the task I just described and ask, when do we get fluorescence from this tracer? So first we had to, first we wanted to simply look across the phases of this task. Now this is a complicated slide. Let me walk you through it. So here's just a diagram of how the expression and the experimental setup is designed. The sensor is infused into the BLA, the fiber is placed over the BLA. And what you can see is that after a very brief period of time during training, we can see quite rapid and distinct transients of fluorescence, which represent acetylcholine binding to the sensor in correct trials and not on incorrect trials. So this is an example of the trace that is Z-scored. And then in this experiment, I'm showing you 4 animals that were subjected to this task. So there's pre-training where they just learn to poke their noses as much as they can. By the time we get to 5 days of training, they're really good at this. Then we change the contingency, they get a timeout. In this particular experiment, these animals did not learn very well, which was very helpful because it gave us much more time to watch the changes in the signal. But what you can see is we have an arbitrary line of 20 rewards achieved in this session that we call acquisition. And each of these individual animals did ultimately cross that line of 20 rewards. And then once they'd all acquired, we now change the contingency, no reward, and they extinguish their response. And across that entire series of days, we recorded all of the fluorescence transients time locked to critical aspects of the task. So either to the receptacle, so the receipt of the reward, the availability of the reward, to the nose poke itself, or to the tone that ultimately predicted the availability of the nose poke for the reward. And you can see that there's some black lines here because we aligned these and so that we wouldn't have overlap between the events. And what you can see here is that each line here represents a day of training with every trial on that day that was successful averaged for one animal. This is data from one animal. And so this is the first, second, third, fourth, fifth day of pre-training. Here is where we change the contingency. These are all the days of training. Here is the arbitrary acquisition of 20 rewards in one session. And then more training days. Finally, here's where we switch to extinction learning. And what you can see in this heat map is that warm colors represent more fluorescence, cool colors represent less fluorescence. And you can see that very quickly, these animals learn that nose poking is something, it's salient, or maybe it's leading to a reward. But what we can see in this trace for this one animal is that there is tightly time-locked fluorescence, acetylcholine fluorescence, to the time of nose poke in all correct trials, or incorrect trials overall. That shifts when we change the contingency and give them a timeout. And for a while, they really aren't, they're nose poking, but nothing's happening. And what you can see is that when they do get a reward, now we get the fluorescence. As the animals begin to acquire the task again, and we see that they're getting better, then we see that the fluorescence shifts back again to the time of the nose poke on correct trials. And as we pass this arbitrary reward acquisition threshold, now we begin to see fluorescence here at the time of the tone that is ultimately predicting whether the reward is going to be available. And you can see less fluorescence here at the time of the nose poke. And of course, here's extinction, where in fact in this animal we saw a nice extinction of the transient to the cue. So this is one animal. In general, what we did then is, because animals acquire at different rates, we only took selected days to do averaging across all the animals. And so what you can see here is the average across animals for the first and last day of pre-training, the first day of training, and then when they got 10 rewards, the day after, the day before they got 20 rewards, the day after, the last day of training, and then I think this is the first day of extinction. And what you can see is exactly the same pattern. So we see time-locked fluorescence on correct trials to the nose poke from very early in pre-training. It gets really tightly time-locked by the end of pre-training, when they're getting, they're really nose poking about, hundreds of times. After we switch the contingency in this experiment, now we lose that signal at the time of nose poke and now get a very nicely time-locked signal to when they do get that reward, which may be surprising at this time. And then it slowly moves back toward the nose poke and ultimately toward the cue, and then begins to extinguish. I'm showing you simply average data, but we also did quite a lot of work to make sure that this was a signal that was significant. And what you can see is, using a bootstrapping method, we saw the same pattern. Those transients were significantly different from the random shuffling of the data. And you can see that it starts at the receptacle even during pre-training, moves to the time of nose poke, moves back to the receptacle, slowly moves back toward the nose poke, and then to the tone. And this dark blue, which I think is actually very interesting and that we haven't yet pursued, but is on our radar screen, is a significant decrease from baseline after receiving reward. And I don't think that's a coincidence. I think this time where we see a decrease in acetylcholine signaling after receipt of reward is likely equivalent to the kinds of changes we saw when we were blocking the nicotinic receptors, and we see a decrease in the activity of this structure. So I think this is potentially also very important for making sense of the cues that are coming in and their behavioral consequence in these animals. So I'm not going to show you the bootstrapping for all of the other experiments, but I can promise you that in fact it looks very similar, that it follows the fluorescent signals that are locked to events. So that was an experiment where we put the fluorescent tracer, the acetylcholine sensor, into the BLA. Can we now find the source of the acetylcholine signal from which those that acetylcholine sensor is actually getting information? And the most prominent input of acetylcholine to the basolateral amygdala is from the nucleus basalis of Meinert, so the NBM. And there are a mixture of chat positive. So chat is the primary synthetic enzyme that results in the synthesis of acetylcholine. Those chat positive neurons can be isolated in chat-Cre mice, so mice that express Cre recombinase under the control of the chat promoter. And so what Rick did was to infuse now a calcium sensor that should reflect to some extent the activity of those neurons into the nucleus basalis, to allow that sensor, that calcium sensor, to diffuse to the terminals in the BLA. He placed a fiber over those terminals in the BLA, and now he asked, do I see a change in the cholinergic terminals in these chat-Cre mice that reflects the signal that we saw with the acetylcholine sensor? And you can see again from this trace during pre-training that again on correct trials, there's a nice transient activation of, or calcium influx into the terminals of these chat neurons in the BLA. It's different from those animals that don't have, performed an incorrect trial. And here I'm showing you two example animals, but this has been replicated in other animals. And again, their behavior reflects the same pattern. These guys learned a bit better because they were, Rick optimized the experiment, so they were in the operant chambers and were much better able to hear and attend to the tone. But you can see during pre-training, they acquire this nose poke really quickly, drops down when we change the contingency and they get a timeout. Then they slowly relearn, they cross this acquisition threshold, then they go through extinction. And what you can see is that here's average data. Again, it starts with a nice a calcium signal in the cholinergic terminals from the nucleus basalis that's time locked first to the nose poke. We see a little bit of smearing here at the reward in these animals that acquired much more quickly, goes back to the nose poke and ultimately back toward when they're getting the tone. We don't have extinction data here because this experiment had to shut down because of COVID. So unfortunately, no extinction data, but all the way up to. So overall, this suggests that, yes, the nucleus basalis terminals have the same pattern of activation as we saw with the acetylcholine sensor in the structure. And that's perhaps not surprising, but it is a nice confirmation that it is the basal forebrain complex that is sending these signals to the basolateral amygdala, and that the change in signal may in fact be upstream of the NBM. So then in terms of neuromodulation, we might expect that the, based on, as I showed you, the complexity of the series of acetylcholine receptors that are present in multiple subtypes of the structure, that if we looked at the principal neurons only and looked at their firing over the same time course, that a neuromodulator may not result in one-to-one driving of their activity. And in fact, that's what we see. So in this experiment, the calcium sensor was driven by a CAM kinase 2 promoter. So it was infused into the basolateral amygdala and was only expressed in neurons that express CAM kinase 2, which is largely the principal neurons. We do see an activity transient. It is not as sharp after in the correct trials and not in the incorrect trials. Here's 3 animals. You can see that they all ultimately passed the threshold of acquisition, although this teal animal did not learn the task very quickly. And what you can see is that here in this example animal, we see a much different pattern of activation. Yes, we see transients of activity of the principal neurons in response to the entry to the receptacle. They're much broader and much more diffuse. This is during pre-training. During training, those magazine entry signals actually get more distinct. They do move back toward the time that the cue is presented, but they're very they're quite a bit less sharp, and we never see a really strong response time locked to the nose poke. Now, this suggests, first of all, that there is not a direct drive of all principal neuron firing by these cholinergic transients. It's perhaps not surprising, since we know from work from multiple laboratories that different subsets of excitatory neurons, principal neurons, may encode different aspects of the task. So if we are recording from the entire group, perhaps that difference in timing and difference in encoding across the population is why we see this diffuse signaling in response to the reward. But if we average across all the animals, actually what we see most prominently is very strong signal around the time of receptacle entry and reward acquisition, some signal here at the time of the cue, although not as distinct as with acetylcholine. And if we compare the two, clearly this neuromodulatory signal is not driving excitatory output of the entire structure, which I think is important for trying to decode what it is that acetylcholine signaling might do in the structure. So now I've shown you a lot of work on observing the acetylcholine dynamics and the change in activity in the structure. What happens now if we change the acetylcholine signaling using an intervention that would increase the firing of these cholinergic neurons? So for this experiment, again, we used chat-cree mice, and in this case, infused channel rhodopsin into the nucleus basalis, the source of those acetylcholine terminals that we measured in the previous studies. That then diffused to, so you can see that there are positive cell bodies in the nucleus basalis. And then we looked for expression of channel rhodopsin in the basolateral amygdala. And you can see that there is expression of the channel rhodopsin in the terminals. It's fuzzy because it's all of those terminals, not cell bodies. It overlaps, although I'm not sure how well you can see that, with the chat staining, which is here in red. And then there was a fiber placed over the basolateral amygdala. And again, we used the same task, but in this case, these cholinergic terminals in the BLA were stimulated, over the entire course of the time that the tone was playing, the previously neutral tone, when this nose poke was active, actually it was the last 2 seconds of the tone, and when the a correct nose poke resulted in availability of the reward. And so first, to show you that this worked, if we did extracellular recordings in the nucleus basalis, there were extracellular transients time locked to the light pulses. And more recently, Rick has shown using dual expression of the acetylcholine sensor in the BLA and channel rhodopsin in the cell bodies of the chat neurons of the nucleus basalis, that there is a nice dose response of acetylcholine release to 1, 2, 5, 10, and 20 light pulses on the terminals in the BLA. So if you look at 0 pulses, there's no release of acetylcholine. 1, 2, 5, 10, and 20 result in more and more acetylcholine release, until at 20 pulses, we see pretty much a sustained elevation of acetylcholine across the time of stimulation. And if he does the same light pulses in an animal that is not expressing channel rhodopsin, but is expressing a fluorescent control construct, he doesn't see any transients at all. So what happens if there is stimulation during the time of, so here's the paradigm, the tone is 10 seconds long. If there's an active nose poke during that 10-second tone, then there is reward delivery for 2 seconds, and the laser is triggered by the correct nose poke and is on for the entire 2 seconds at 20 hertz, that the reward is available. And so what you can see here is that during the pre-training, when the animals are simply nose poking constantly and getting rewards every time they're available, there's no effective stimulation. And that's actually consistent with a fair amount of data that suggests that the FR1 responding for a reward is not very sensitive to BLA activity, and particularly to BLA plasticity. But now, once the contingency is changed, now you can see that the unstimulated animals do learn this task very nicely. There's the reward threshold. And that the animals that are stimulated whenever they make a correct nose poke learn the task significantly faster. And one thing that we were quite surprised about is that there was no difference in extinction in the 2 groups of animals. And that was different from the fear conditioning experiment done by the Roll and Talmadge labs, where in fact they didn't see a very strong effect of stimulating during fear learning. They saw more of an effect of prolonging extinction. So we already have a distinction between the fear cue learning and the reward cue learning when we stimulate the acetylcholine neurons. And I want to show you here the incorrect nose pokes, because from this design, we cannot distinguish between a role for acetylcholine in allowing the animals to withhold incorrect responding, or a role for acetylcholine in encoding the correct responding. So there is a tension here, because what you can see is there's, as I told you, 400 nose pokes during the pre-training when there's no cost to nose poking. It's just phenomenal. They're just sitting there and poking their nose. And there's no difference between the acetylcholine and the stimulated and the unstimulated animals. So that does suggest there's no effect on activity. There's no effect on motivation. There's no effect on overall nose poke ability. But now here, when they're learning the contingency, you can see that the stimulated animals come down more quickly in their incorrect responses. And of course, the control animals do come down as well, just more slowly. So that's to be determined. Is it because acetylcholine is really good at helping them inhibit inappropriate responses, or to encode appropriate responses? And we're working on that now. So here is another surprise that came from a control experiment. And I actually had an interaction online with Nadia Chowdhury, whose work I'll talk about in a second. It is the unexpected response to a control experiment that sometimes is what shifts your understanding. So in this experiment, what Rick wanted to do is to have a group that was stimulated the same number of times. So they got the same number of light pulses, they got acetylcholine release, but they were explicitly unpaired from any of the events, not nose poking, not reward availability or acquisition, and not the cue. So those 2 animals, sets of animals, got stimulated same amount, but now they either had the acetylcholine during the time where we thought that perhaps the acetylcholine signaling would be essential for linking those events, or in a time that was between those events. And what you can see is that the two groups who were stimulated either paired or explicitly unpaired saw the same increase in or acceleration of learning across the course of this experiment. So here's the unstimulated group. Here is the light blue is the yoked group, and dark blue is the explicitly paired group. So that was confusing, but as I mentioned, oh, and similar for the incorrect nose pokes. As I mentioned, this actually is reflective of some data that had been really revolutionary in the field of nicotine addiction. And that was, again, a surprising set of responses in rats that were learning to self-administer nicotine. And what Nadia Chowdhury and her collaborators found was that rats will nose poke a little, or will press a lever for a mildly reinforcing stimulus, which is just a light. And I don't know if you have ever had a 2-year-old, but even though turning a light on and off is not particularly rewarding, it's still salient, and kids will do it. So the same is true for rats. They will press a lever to change the light, to turn it on and have it turn on and off. But what they found is that if that light was contingent with the delivery of nicotine in a self-administration experiment, they got an increase in self-administration, consistent with the idea that nicotine itself is reinforcing. And that's the design of most self-administration experiments. You have a tone or a cue or a light that predicts availability of the drug. You press a lever and you get the drug. But in this experiment and in others, it's been replicated many times, non-contingent nicotine increased responding for the cue just as well as contingent nicotine, suggesting that overall what nicotine did in this experiment was not to simply reinforce the association between the cue and the drug, but in fact to enhance the basically the rewarding efficacy of a mildly reinforcing unpaired cue. And that for the nicotine field is really important. So when do you smoke? When you've had a good meal. In fact, right after you've had a good meal. People say that it enhances their enjoyment after other rewarding stimuli, in a bar after a drink. And overall, this role of nicotine to increase the rewarding value of mildly reinforcing stimuli may be really key to understanding why people continue to use nicotine products. And so one of the questions that asks is that in our experiment, are we simply seeing a rewarding effect of the light itself? And so Rick did a couple of different controls. One is to just see if animals would poke for laser stimulation in channel rhodopsin animals. He did not see any maintenance of behavior by the light itself. And he also did a real-time place preference experiment where the animal was stimulated only in one side of a 2-chamber box. And you can see that animals did not explore any more time in the chamber where they got light stimulation, even over 2 days of training. So we aren't seeing a primary reinforcing or rewarding effect of the light itself, but we may be seeing either a consolidation of the plasticity or an increase overall in the efficacy of these mildly reinforcing cues. So then that brings us to the question of, is nicotine involved? Are we reflecting, or are we showing some effects of nicotine? So we have some unpublished data on nicotine. It doesn't appear that nicotine has quite the same effect as light. So there is a nearly significant effect on correct responses, but it's not very impressive. There's a slightly better effect on incorrect responses. Nicotine can decrease significantly the number of incorrect responses over this timeout learning period, but it doesn't really look like acetylcholine. So if we then do the experiment, but in the presence of a nicotinic antagonist or a muscarinic antagonist, or the two together, can we see an underlying role for acetylcholine signaling in this task? And so in this experiment, we have the same design, but now either a nicotinic or a muscarinic antagonist is given just before each session during pre-training and during the timeout training. And this was these were experiments done by Kristen Kim when she was a rotation student in the lab. She's now a graduate student in the lab. And what you can see here is, again, another surprise. The nicotinic antagonist had no significant, well, none of the antagonists had any significant effect during pre-training. Again, no motor effects, no inability to learn an FR1 response for reward. That's all good. But in fact, during the time when the timeout actually was really important and the animals had to learn that they could only poke during the time that the tone was playing, you can see that those animals got a muscarinic antagonist alone or got the muscarinic plus the nicotinic antagonist, could not learn this task at all. The nicotinic blockers didn't really do any, there's no significant effect here of blocking the nicotinic receptors. So overall performance of this cue reward learning task during the timeout conditions definitely requires muscarinic receptor signaling and doesn't need nicotinic signaling. So it may be that, in fact, the way that we've designed this pharmacological experiment is revealing more of an effect on consolidation or other aspects of the task that are linked to plasticity that is not time locked to the cue reward pairing. And I can tell you that some experiments that are ongoing suggest that giving the muscarinic blocker after each session also can change the responding on this task. So there may be an effect for plasticity that continues after the task. But at least for now, I would say that the nicotinic signaling is not the driver of the plasticity in this task, which is disappointing, but there are other things that we can look at to explain the reward enhancement. So that's where I'm going to stop. I'm going to conclude with just some very basic summaries. We know that acetylcholine is released in the basolateral amygdala to task relevant events. It shifts after learning. The pattern looks a lot like prediction error signals that you get in the dopamine system, in the VTA and other neuromodulators. However, the fact that you can stimulate acetylcholine non-contingently or completely explicitly unpaired to relevant events and get an improvement in learning this task suggests that this is not required, that a reward prediction error signaled by acetylcholine is not required for correct performance of this task. So I think we need to look elsewhere. I think upstream is probably a reward prediction error signal, maybe the dopamine signal, maybe other signals. But I don't think that's what the acetylcholine is doing. I think it is triggered by salient events, results in a change in neuronal signaling that initiates processes of plasticity that are long lasting, and that change in plasticity is probably what's more important for its effects. I showed you that acetylcholine release does modulate activity in the structure, but it doesn't drive BLA principal neuron firing. Those patterns of activity don't match, even though overall tonic acetylcholine is essential to setting some degree of firing of the structure. And then finally, I showed you that stimulation of the nucleus basalis to BLA chat positive terminals in the BLA is permissive for learning. It potentiates appetitive learning, but different nicotinic receptors are involved in this appetitive task than are involved in the plasticity in the fear conditioning experiments. They're blocking nicotinic acetylcholine receptors, particularly alpha-7, decrease the plasticity. Here, the muscarinic receptors appear to be really essential, and there's more experiments to be done there. So I'm going to stop there. I'd like to, again, thank the people and acknowledge the many people who contributed to these experiments, particularly Eric Girardi, who I didn't mention, who just had a birthday. Rick really did design and carry out the reward learning experiments, partially because he has a passion for learning and memory experiments. And so putting together the interests in the lab around acetylcholine and his passion for learning and memory were essential to getting these done. Kristen Kim really stepped up and did some beautiful experiments as a rotation student with many, many other things going on. So it was great to see her join the lab. I mentioned Jan Miner's work, and Jan is right here. We've had collaborators who are critical to this work, including particularly Yulong Li, without whose sensor, none of this would have been possible. I want to thank our funders, without whom, again, this work wouldn't have been possible. This is an earlier iteration of the lab before we had to wear masks. Please come take a look at our website or follow me on Twitter, and I'm happy now to answer any questions that may come up. Thank you very much for your attention.
Okay, thank you so much. It's been a great talk, lots of impressive data. So Christina, why don't you start asking questions?
Absolutely. So the first question we have is, during extinction, was there a significant increase in the acetylcholine signal around the time that the animal expected a reward to be delivered?
Yeah, so the question is, was there a transient at the time of the expected reward? And the answer is not much, no. We didn't all of a sudden see that signal come up. I suspect that means that you had to actually get the reward to get the signal during the early timeout learning. So in those traces that I showed you, those are correct trials. We don't see a lot of activity in incorrect trials, which is interesting. You'd think that if it was a prediction error, that we would be seeing, even in the incorrect trials, something in response to a nose poke, something in response to the tone. We don't see it there, and we don't see it in extinction. So there's maybe something more related to the primary now conditioned reinforcement of, so first there's the primary reward, then there's conditioned reinforcement of the nose poke and then of the tone. So great question.
Yeah.
Second one.
Christina, your voice is very low. Maybe you
Very
can
low.
be closer to the microphone.
Yeah, I'll try. Is this better?
Almost.
Okay. Sorry for that. Yes, here. It's at the
That's better.
maximum. Sorry.
Okay.
It's better. We hear you. That's okay.
Okay. Second question. Do you think that cholinergic stimulation during extinction might alter the time course? Not sure about the stimulation paradigm or what direction it might shift.
Right. So for the fear conditioning experiment, it seemed to alter the time course of extinction. It definitely delayed extinction. In our hands, we have not seen any shift. Nothing budges extinction in our hands. The animals that got the muscarinic antagonist throughout training really never acquired the task, so we don't see a high enough, baseline performance to see a decrement. That's the only time where we saw less extinction response, and that's because they didn't get it. They never learned the task.
Yeah. Great. Third question is, how do acetylcholine sensor fluorescence increases evolve over the course of a single session across multiple trials during one day?
And that's a fantastic question. And I will add a different question also, and that is, how does the performance of the task evolve across single trials when you get the non-contingent stimulation in between them? And the answer to both of those is we have the data and we don't know yet. We would like to actually ask both of those questions, and I don't have an answer for you.
And lastly, how do you integrate dopamine signaling in the way acetylcholine is assigning balance to a cue? What outputs of the BLA do you think are important in these specific processes you presented?
Yeah, I think that's the key question. And there was one study in Current Biology that suggested that the source of the acetylcholine inputs may define whether the output of the BLA or of the amygdala, whether it's central or basolateral, was rewarding or aversive, and that we don't see based on the experiments that we've done so far. Another possibility is that overall, it's not the acetylcholine that assigns the valence, but it in fact increases the likelihood that an event, whether it is aversive or appetitive, will result in pairing with the output, so that the likelihood or the assignment of valence is occurring through various populations of principal neurons. All of those principal neurons are probably getting the acetylcholine input. And overall, it's whether or not you have the output reinforced to for example, the VTA or nucleus accumbens, or to brainstem structures that are more involved in threat, that will result in the cue outcome valence. It doesn't appear to be acetylcholine as far as we can tell. which I hoped it would be, but it's not. I don't think.
So it seems that muscarinic transmission is important in the phenomenon that you described. So what do you think on a cellular level or on a synaptic level is happening in the BLA?
So what I think is likely happening is that the muscarinic signaling is resulting in increased calcium signaling in neurons that are encoding the cue or that are downstream that are the output neurons. So that overall, that calcium signaling is prolonged by the muscarinic stimulation. It results in perhaps a longer window for LTP. So that could be happening during the event, but it clearly outlasts the short time scale of stimulation, because we see it even when we do non-contingent stimulation. And then I think and this is consistent with a very old pharmacological literature, the role for acetylcholine signaling in events that are occurring for 3 or more hours after learning, so during the consolidation phase, are also likely then a consequence of that intracellular signaling persisting, likely getting to transcription factors that are beginning to initiate long-term LTP events, and that if you have enough stimulation of acetylcholine during the time when you are initiating those signaling pathways, that you get to the nucleus rather than, stopping at the synapse.
Okay, thank you. Christina, you have more questions or?
I do not. Oh yes, there is one. For the acetylcholine sensor fluorescence, does it make it so the muscarinic receptors don't work? And would that have an effect on acetylcholine in the BLA?
Great question. So you could imagine that if the sensor is super high affinity and you have lots and lots of now artificial muscarinic receptors expressed, you might soak up all the acetylcholine and result in a change in behavior, right? So I think that's still formally possible because we haven't looked at it directly, but we don't see any effect of expressing the sensor itself on the time course of learning. So I don't think it's shifting that learning in any very measurable way. Could be subtle. And also I think that that's a factor of it being a tracer. It's still present at fairly low levels. The affinity for acetylcholine is not higher than the endogenous receptors. And just as in human imaging, PET imaging studies, it's a reflective of the overall activity and at low enough levels that it's probably not interfering. But formally, we cannot rule it out.
Yeah.
Okay.
All right.
Cool. That was inspiring. And for us too, these genetically encoded transmitter sensors, they really are a game changer. I think they give you temporal resolution and they make it more easy for many labs to look and visualize what the transmitter is in a specific task. It's you've done this very nicely for acetylcholine.
Thank you.
Cool.
Well, thank you for the great session, and thank you for the questions.
Yes. So we're almost to the hour, so we're gonna close the talk here, and please come back next Thursday. It will be Manuel Mameli from Lausanne, hosted by Eric, and a co-host that we still have to find. Okay, with that, let's conclude here. Thank you so much and have a nice rest of